We have 6 presenters for this oral presentation. All right, all presenters have been promoted. Before we begin, I would like to introduce our judges for today for this session. The first one is our respected speaker who just concluded his talk, Dr. Alan Yu from Monash University, Malaysia. And we are also honored to have Professor Dr. Nafiza Haji Muhammad Ismail from International Medical University, who is also the president of MSPP and also the co-chair for this event. So thank you very much, Dr. Alan and Prof, for being with us. We will begin with our first presenter. All right, just as a reminder, for all presenters, please stay on after the 7 minutes presentation, as you will be having a live Q&A by our judges. Without much further ado, I'm presenting to you the first presenter for today, Mr. Omchit Surien.
Hello everyone, my name is Omchit Surien from Universiti Kebangsaan Malaysia. So today I'm going to share my research work entitled Histopathological Analysis of Psilocybin as Chemopreventive Agent Against Lung Squamous Cell Carcinoma in Mouse Model. So for the introduction, lung cancer is divided into few types based on the histopathological observation. So in this study, we are focusing specifically on squamous cell carcinoma, which is one of the common type of human lung cancer. So the lung SCC or lung squamous cell carcinoma is defined as the abnormal growth of the cell from the bronchial epithelial cell to the hyperplasia, metaplasia, and lastly we progress to become a cancer or SCC. So other than histopathological observation, lung SCC also can be differentiated from other type of lung cancer through the expression of cytokeratin 5-6, where usually lung SCC tumor has the overexpression or highly expressed cytokeratin 5-6. According to the latest statistic, lung cancer is one of the common type of human cancer, and lung cancer is leading cause of cancer-related death, where the high mortality rate among lung cancer can be contributed by many factors, and one of them due to the late diagnosis, where most of the lung cancer patients were diagnosed at the advanced stages. This late diagnosis will increase the risk of treatment failure that lead to the high mortality. So the chemo prevention is one of the alternatives to the treatment to reduce the mortality rate, and the chemo prevention is defined as the use of any natural, synthetic or biological agent to delay, suppress or reverse the carcinogenesis. So in this study, we investigated the chemopreventive effect of psilocybin against lung SCC. So this psilocybin is a natural compound that can be found in the grape, blueberry and also tree wood of Tecopus marsupium. This psilocybin is a potential to be developed as a chemopreventive agent because of psilocybin has been proven to exhibit the various pharmacological properties such as anti-inflammatory, antioxidant, anti-proliferation and also anti-angiogenesis and metastasis. Moreover, this psilocybin also has been proven to exhibit the anti-cancer against lung SCC cell line in vitro and also in vivo mouse xenograft model. However, the chemopreventive effect of psilocybin against lung SCC in chemically induced mouse model has yet to be determined. So this is my research question and my research hypothesis. So for the general objective, to investigate the chemopreventive effect of psilocybin against lung SCC in mouse model. And the specific objective, my first specific objective to determine the effect of psilocybin on lung SCC through the histopathological observation and also thickness of bronchial epithelium layer. And my second specific objective to determine the effect of psilocybin on semi-quantitative histopathological scoring. And my third and also last specific objective to determine the effect of psilocybin on the protein expression level of cytokeratin 5-6. So next, I move on the methodology. So in this study, the total of 24 female biopsy mice were randomly divided into 4 groups. So for the first group is the vehicle control group, where this group treated with the corn oil through the intraperitoneal injection that act as a vehicle for the psilocybin, and 70% of acetone that act as a vehicle for the carcinogen through topically at the shaved subscapular area. And the second group is the cancer group, where this group we treated with the carcinogen NTCU to induce the lung SCC. And the third group is the low dose of tocilizumab, where this group treated with the 10 mg per kg of tocilizumab together with the carcinogen. And the last group is the high dose of tocilizumab with the 50 mg per kg of tocilizumab together with the carcinogen. So after 26 weeks of the treatment with the twice a week, all mice were sacrificed and the lung were collected for the test. So for the objective one, we proceed with the H&E staining, hematoxylin and eosin staining. For the histopathological observation under the light microscope. And for the H&E staining also, we measure the thickness of epithelium layer using the ImageGFG software. And for the histopathological semi-quantitative scoring, is based on the pre-malignant stages in the development of lung SCC. And for the objective 3, immunohistochemistry was used to investigate the expression level of cytokeratin 5/6. And the quantification of cytokeratin 5/6 also was done using the ImageGFG software. So next, I move on the result and discussion. So for the first part is the histopathological observation. So as we can see here, the vehicle control group show a normal characteristic of lung histology without the thickening of the bronchial epithelium layer, and the high magnification is show the normal characteristic of bronchial epithelium layer with a single ciliated columnar epithelium and basally located nucleus. In contrast, the group that received the carcinogen without tocilizumab show the thickening of the bronchial epithelium layer, and at high magnification it show the disorganization of the cell with the increased nucleus cytoplasm ratio and also the formation of keratin pearl. And the low dose of tocilizumab only developed the hyperplasia at the bronchial epithelium layer, and the high dose of tocilizumab still maintain the normal characteristic of bronchial epithelium layer with a single ciliated columnar epithelium that's similar to the vehicle control group. Next, we look at the histopathological scoring. So as we can see here, the cancer group scored the highest, with the mice in this group usually develop dysplasia and SCC in the lung. And the tocilizumab reduced the histopathological scoring, with the most of the mice develop the hyperplasia and still maintain the normal bronchial epithelium layer. So next, we look at the thickness of bronchial epithelium layer. The cancer group or NTCU significantly increased the thickness of bronchial epithelium layer, but tocilizumab treatment significantly reduced the thickness of bronchial epithelium layer. And this increasing in the thickness of epithelium layer related to the severity of pre-malignant stages in development of SCC. Lastly, we look at the protein of expression level of the cytokeratin 5/6. So we can see here the cancer group significantly increased the expression of cytokeratin 5/6, but tocilizumab significantly reduced the cytokeratin expression level of cytokeratin 5/6. So from our finding, we conclude that tocilizumab was able to prevent the development of lung SCC in mouse model. Therefore, the further investigation is needed to understand the underlying molecular mechanism of tocilizumab as a chemopreventive agent. So this is my references. Acknowledgement. Thank you.
Thank you very much, Mr. Omchit. We move on to the Q&A. So what we will do is we will have only one question for each presenter, and we will alternate the judges. So I think I will have Dr. Alan first to ask question to our presenter, Mr. Omchit.
Right, so yeah, thank you. So it's a very nice presentation by using the animal model. So I just have a quick questions regarding the cytotoxicity of this particular drug. So it seems that it's quite potent, right, in terms of curing the cancer. But the thing is whether this particular drug will also cause cytotoxicity as well.
Okay, thank you, Doctor, for question. So basically, this compound, which is tocilizumab, is a well-studied compound, and the toxicity study also show that this tocilizumab is quite safe, even though it's given at a high dose in the clinical and also the preclinical. They also have been tested in the human, where this tocilizumab also has been safe to consume up to 200 mg per kg. And in the animal, they try the high dose as well, which is, if I'm not mistaken, is almost 1000 mg per kg, and it still show no toxicity or adverse drug effect.
Okay, it's good to hear that. Thank you very much.
All right, I think that is all. Yeah, Dr. Alan, thank you, and Mr. Omchit, well done. we will move on to our next presenter, Miss Ramya. Are you there, Miss Ramya?
Yes, I'm here.
All right, can we have the video ready?
Yeah.
Okay, we'll have Miss Ramya Dewi from Universiti Kebangsaan Malaysia.
Good morning. I would like to present my project and a very good morning. I would like to present my project entitled Effects of 1,4-benzoquinone on DNA repair pathway and detoxification enzymes targeting hematopoietic stem or progenitor cells of different lineages. Benzene is categorized as most stable aromatic hydrocarbon, which is found in industrial solvent, and it has been categorized as common environmental contaminant. It has been found in the environmental in which humans are exposed to benzene through the automobile exhaust, smoke, and also gasoline vapors. Yes, benzene is toxic because it has been Group 1 in International Agency of Research Cancer, in which chronic exposure of benzene has been reported in bone marrow depression and many types of leukemia. And then it has been reported that benzene has to be metabolized in order to produce reactive metabolites known as phenol, catechol, hydroquinone, and benzoquinone, which play an important role in benzene toxicity towards the hematopoietic systems. Overall, the metabolism of benzene involves two pathways which happen in liver and also in bone marrow. In liver, they have metabolic activation to produce phenol, which are then metabolized by the MPO, also known as myeloperoxidase enzyme and NTO1 enzyme, to produce 1,4-benzoquinone and hydroquinone, which are very reactive, and it has been reported to induce hematotoxicity and leukogenesis. What is hematopoiesis? Hematopoiesis consists of a group of blood cells which are known as the progenitor cells, which actually proliferate and also differentiate in order to produce different types of mature blood cells. And the balance in hematopoiesis is very important in order to maintain a balanced system of production of mature blood cells, because if there's anything in regulation or any chemicals, it actually can cause production of leukemic stem cells, also known as cancer blood stem cells. Actually, benzene hematotoxicity and leukogenesis have been reported to occur via multiple mechanisms which are involved: chromosome aberration, oxidative stress, alteration of gene expression, error-prone DNA repair, and also epigenetic regulations. DNA damage can play a fundamental role in the hematopoietic system function, because if there is the repair have occurred but is not repair, it can cause the stem cell to proliferate in order to produce stem cell containing the mutation. And if the DNA repair occur in an error-prone way, that means the misrepair occur. This also can cause mutation in the stem cells. Thus, it's very important to study the DNA repair system occurring in the hematopoietic system. Apart from that, the double-strand breaks that caused by the benzene actually cause the DNA repair, which is error-prone, which also play an important role in the benzene-induced hematotoxicity. This DNA repair is divided into two main groups, which are known as homologous recombination and non-homologous and joining repair. But according to the report previously, both actually are involved in the genomic instability. It is very vital for you to study the benzene toxicity in the hematopoietic system complex, because hematopoiesis consists of niche. It's a dynamic microenvironment consisting of various types of progenitor cells which play an important role in production of mature blood cells such as lymphoid, myeloid, and also progenitor cells. Apart from that, previous studies also reported there is a lineage direct toxicity of benzene, which actually occur differently according to the different type of progenitors such as myeloid, lymphoid, and also erythroid. Thus, it's very important to look at this lineage directed strategy. The experimental design actually starts with the sacrifice of an elder male mouse in order to obtain the bone marrow cells, which is actually cultured for 24 hours in vitro, and it is exposed to various concentration of the 1,4-benzoquinone before it has gone through the colony-forming unit assay to produce respective progenitors such as lympha progenitors, myeloid progenitors, and also erythroid progenitors. Okay. And then it is subjected to DNA repair profile and also detoxification profile, which involve DNA PKC enzymes RAD51 for the DNA repair profile and MPO and also NQO1 for detoxification profiles. The results shows that there is actually differential responses among the progenitors in which, for example, for non-homologous end joining results reported to be an error prone actually happen most probably in the myeloid cells, as you can see it occur even starting from the lowest concentration of the 1,4-benzoquinone. For RAD51, which represent the homologous repair, it is not actually significant in erythroid, but it is shown significant in myeloid and pre-B, but it starts as lower as the 5th concentration of 1,4-benzoquinone. For detoxification profile, myeloperoxidase, this shows that this is one of the metabolic enzymes which play an important role in benzene toxicity. And from here, we can see that the myeloid play an important role as well. Okay, and then for NQO1, which plays a detoxification enzyme, it shows that myeloid happen to be the most sensitive for the reduction of the NQO1 enzyme. Therefore, the promotion of error-prone is actually very important in the genomic and stability via chromosomal deletion, insertion, or translocations. Apart from that, the MPO and NQ1 also are very important in the BQ and HQ redox conversion benzene toxicity. So in conclusion, we can conclude that 1,4-benzoquinone exposure causes error-prone DNA repair and reduced detoxification, which varies among the progenitors, and we can conclude that it is lineage dependent. Thank you.
All right. Okay. Thank you very much for the presentation. I will invite Professor Dr. Nafiza to ask questions.
Thank you, Dr. Izuddin. Ramya,
Yeah.
are you known as Ramya or Dewi? Which one do you prefer?
I prefer Ramya.
Ramya,
Yeah.
all right.
Yes.
You've done a lot of work. I can see that, and
Yes.
you were sort of going through all the work that you've done to make your presentation as comprehensive as possible.
Yes.
My question to you is, what prompted your interest to do this work?
Means that Prof telling what makes me interested to do this research in terms of DNA repair. Okay,
no, in terms of in terms of I think if I'm not wrong, you started off by saying that benzene causes hematopoietic cancers.
Yes.
Right, in humans.
Yes, correct.
Yeah. So what prompted your interest?
Okay, why I do this study on the hematopoietic stem cells cancers, because nowadays we are having exposure to the environmental, for example, like benzene, which is very important as a hematopoietic stem cell inducer, cancer inducer. Apart from that, we have leukemia. Most of the treatment is targeted on the whole population of the hematopoietic stem cells, rather than according to the lineage in terms of therapy. So I hope that my research actually can open a pathway to understand the genetic mechanism, which is different, which is seen different according to the different lineages, so that we're able to target only at one lineages rather than comprehensively the whole part of the hematopoietic stem cells. So this actually in order to open a new pathway in terms of pharmacological development in terms of cancer treatment.
Okay, whether benzene induced hematological cancers or not in humans, is it a causation or is it a correlation?
It's a causation, yeah.
How do you,
causation. More to
yeah, can you elaborate on it? It's interesting if you say it's a causation. Why do you say it's a causation?
It's a causation because benzene has been one of the model that has been proved to cause hematopoietic stem cell in terms of these metabolites. So these metabolites has been reported in order to have a genetic and also epigenetic modulations. So we can conclude that this benzene actually can cause this hematopoietic stem cancer in terms of causation.
Are you saying that a lot of hematopoietic cancer patients, studies have shown that they are all exposed to benzene, or most are exposed to benzene?
Yes, most are exposed to benzene because most of the studies actually involve the occupational workers who work in the industrial companies related to the benzenes. Benzenes are such as paint and also oil industries and also glove companies. They're actually very exposed to benzene. And they do the tests in terms of blood tests and also genetic tests. They found that there is a lot of genetic mutations, especially the chromosomal instability that is found in the workers exposed to this benzene.
Yeah, thank you, Ramya. But this is to provoke further your interest. That if they are exposed to benzene, if we say when they work in glove factory, they're exposed to benzene. They're also exposed to a few or maybe hundreds of other chemicals. So to prove causation in humans, especially, is very difficult. But thank you. Thank you.
All right. Thank you. Okay.
Thank you so much, Ramya and Prof. I believe there's one question in the Q&A chat box for Ramya. Appreciate if you could type in the answer in the Q&A chat box. Yeah, thank you, Ramya. We will move on with our next presenter, Ms. Harishini Rajaratinam. Are you there, Ms. Harishini?
I'm here.
All right, so Ms. Harishini is from Universiti Science Malaysia. So please take it away. Thank you.
Hello everyone, my name is Harishini Rajaratinam and I'm from Universiti Science Malaysia, USM. My paper ID number is OCA6 and the title of my study is The Influence of Breast Cancer Therapy on the Expression of Anti-Neonatal NAD 1.5 Antibodies in the Serum of Breast Cancer Patients. I'm truly honored to be part of this conference. The expression of neonatal NAD 1.5 has been known to potentiate metastasis in breast cancer. The 5' neonatal NAD 1.5 is the splice variant of the LDAR isoform. The distinguishable 7 amino acid changes has been highlighted in figure 1b. The location of the splicing generally takes place at the segment 3 and segment 4 of domain number 1. The electrophysiological changes caused by this alternative splicing would modify the kinetics of neonatal NAD 1.5 channel and thus allow a greater intracellular sodium current to take place. How does this particular sodium current contribute to invasion and metastasis? The influx of sodium current will actually contribute to the efflux of hydrogen ions. The accumulation of these hydrogen ions would lead to the change in the pH, where acidic pH would contribute to the activation of cathepsins or proteases in order to release proteolytic enzyme that is very useful in order to degrade the extracellular membrane and thus ultimately contribute to invasion and metastasis. For breast cancer treatment can be divided into two categories. One is local, which includes surgery and also radiation therapy, and the other one is known as systemic, that includes hormone therapy, chemotherapy, and also targeted therapy. In recent days, a new form of therapy is being included, which is known as immunotherapy. In March 2019, FDA has approved a medication known by the name atezolizumab, which acts as an immune checkpoint inhibitor in order to treat triple-negative breast cancer. This year, a paper was published in the Frontiers of Pharmacology, which highlights the functional role of NAD 1.5 in breast cancer metastasis. A particular point caught our attention, which suggests that is it possible to make neonatal NAD 1.5 as a biomarker for early diagnosis of breast cancer. We decided to incorporate the idea and propose a new concept whereby we would like to incorporate immunogenicity of neonatal NAD 1.5 as an immunosurveillance marker in order to monitor treatment efficacy. Till date, the discussion on neonatal NAV 1.5 has been restricted to its role in potentiating breast cancer metastasis through studies that utilize breast cancer tissues and also cell line. However, its immunogenicity, which is reflected by the production of antibodies against neonatal NAV 1.5, has not been studied yet, and let alone the immunosurveillance function of these novel antibodies. Let's proceed to objectives. So my main objective is to decipher the influence of breast cancer therapy on the expression of anti-neonatal NAV 1.5 antibodies in the serum of breast cancer patients, followed by 3 specific objectives. So my first specific objective is to compare the expression of anti-neonatal NAV 1.5 antibodies between healthy participants, pre-treatment breast cancer patients, and also ongoing treatment patients. My second specific objective is to elucidate the presence of metastasis via cytokine analysis for each 3 groups mentioned earlier, and followed by the last specific objective, which is the correlation between the first and the second specific objectives. In this study, a total of 96 participants were divided equally into 3 groups, which are healthy participants, pre-treatment breast cancer patients, and also ongoing treatment breast cancer patients. The study took place at Breast Cancer Awareness and Research Unit in Hospital Universiti Science, Malaysia. The inclusion and exclusion criteria for each group has been included in the slide. The separated serum was subjected to in-house ELISA in order to detect the presence of anti-neonatal NAV 1.5 antibodies, followed by cytokine analysis. Since anti-neonatal NAV 1.5 antibodies are quite novel, and therefore we could not find a commercial ELISA kit in order to detect the presence of these antibodies. Therefore, we decided to develop our own in-house indirect ELISA assay. Let's have a look at the results. Based on the comparison made, the pre-treatment group exhibited the highest expression of anti-neonatal NAV 1.5 antibodies, and most important of all, there was a significant difference between the pre and the ongoing treatment group. The cytokine analysis revealed the downregulation in the pro-inflammatory cytokine in the ongoing treatment group, which was marked by the red arrows, and this signifies the interrupted presence of metastasis under the influence of breast cancer therapy. In addition, there was a significant positive correlation between anti-neonatal NAV 1.5 antibodies and interleukin-6 in the pre-treatment group, followed by a significant negative correlation between VEGF cytokine and also anti-neonatal NAV 1.5 antibodies in the ongoing treatment group. In this study, we found that the presence or the regulation of anti-neonatal NAV 1.5 antibody is consistent with the presence of metastasis in the pre-treatment group, and such scenario is completely reversed in the ongoing treatment group, where the downregulation of these novel antibodies suggests the treatment efficacy or the influence of breast cancer therapy. The consistency in the expression of anti-neonatal NAV 1.5 antibodies and its significant correlation with well-known cytokines highlights its potential as an immunosurveillance marker. Therefore, such functions should be exploited in order to monitor treatment efficacy and also to detect early progression of breast cancer. As simple as using blood samples, we are able to carry out such act. In conclusion, the presence of anti-neonatal NAV 1.5 antibodies highlights the immunogenicity of neonatal NAV 1.5, and the expression of these novel antibodies is influenced by the breast cancer therapy. And that's all from me. Thank you very much.
Thank you, Ms. Harishini. I'd invite Dr. Allen to ask any questions.
Okay, that's good. Yeah, so it's a very nice talk. So I think the findings are actually very interesting. So just have a couple of questions regarding this antibody. So is this antibody a monoclonal antibody, and how specific it is towards the neonatal NAV 1.5? And the second question is, this antibody, is it only good for detection, and whether this antibody would have any, say, function blocking properties? Okay, thank you.
Okay, thank you, Dr. Allen. So for the first question, how do I
how does it pay attention on the specificity? This is because the coding of the ELISA assay has been conducted using a peptide that was designed specifically to mimic the 7 amino acid changes in the neonatal NAD1.5. So the binding basically has already been detected through our peptide sequence and so on. And for the second question, yes, these particular antibodies, they have properties, like pharmacological properties. There's a study, doctor, basically, I think in the year 2019, there was a study conducted using some sort of this kind of antibodies, which is also part of neonatal NAD1.5. It's just that it's a different specificity. So they use it in order to suppress the detection of ovarian cancer. So our future prospect is that we would like to use this particular novel antibodies in order to emerge as an immunotherapeutic target in order to reduce the progression of metastasis.
Okay, that's good. Yeah, so hope that you will be able to make the best use of this antibody in terms of treatment. Okay, thank you.
Thank you, doctor. Thank you.
Okay, thank you, Harishini, and thank you, Dr. Ellen, for the questions. We will move on with our fourth presenter. Ishpa Shapiah Abdullah from Universiti Brunei Darussalam. Are you there?
Yes, I'm here.
All right, so we'll have the video ready. All right, go. Thank you.
Hello, I'm Ishpa Shapiah Abdullah, a pharmacy student from Universiti Brunei Darussalam. The topic for this presentation is on over-the-counter medicine attitudes and knowledge among university and college students in Brunei Darussalam. In this presentation, I'll be talking about the introduction, methods, results, discussion, conclusion, and lastly, I will show the references. Introduction. What is self-medication? It is an act where a person can purchase the medications to treat their symptoms without any consultation from a healthcare professional. Globally, inappropriate self-medication with OTC has been increased and recognized as a public concern. A worrying trend reported that misuse of various painkillers, vitamins, and sedatives was found among university students. According to the database in Australia, paracetamol is the most frequently used in overdoses with 108% increase in acute liver toxicity. Across Southeast Asia, the self-medication practice with OTC is very common, which can be seen in Brunei also. Although Brunei offers free medical treatment and medication, the general public sometimes practices self-medication for minor illness. Most of the time, they purchase OTC according to the knowledge and experience on the effectiveness of the medication. A documented Bruneian study assessing the use of OTC medicines is lacking. So the younger population, especially those undergoing university or college education, are the primary user of social media that are likely exposed to unverified sources of health information. Also, with the advance of the technology, the internet usage entered into the daily lives of higher education students is rapidly increasing. So the aim of the study was to examine the safe use of OTC by determining the knowledge, attitude, and practices of OTC among the public university and college students in Brunei. Methods. This study was an online survey which was conducted from March to April 2021. So the targeted participants were Bruneians or permanent residents of full and part-time students from all faculties in public universities and colleges. So the questionnaire was adapted from a survey conducted by Tesfa Mariam with additional information from US FDA on the OTC medicine for consumer. So the questionnaire was sent to the students' official email address with a reminder of 2 weeks interval. Our software and Microsoft Excel were used for data analysis. So the pilot test was conducted among 38 students. Results. 335 respondents returned a completed questionnaire. The characteristics of the students can be seen in this table. Association of demographic characteristic with level of knowledge. If the p-value is less than 0.05, it means there's a significant difference. In this case, the academic degree and the course of study shown a significant difference among the groups, with a p-value less than 0.05. Attitude. Almost all the respondents indicated a positive attitude towards the use of OTC. Practices. When asked if they have ever experienced adverse effects before, 9.9% have ever experienced it before, while 17% do not know. I also added a question, what did they do after experiencing the ADR? They either go see a doctor, sleep, or do not do anything. When asked if they have ever taken more than recommended dose, 8.7% have ever taken more than recommended dose, while 7.8% do not know. I also added a question, which OTC medication did they have ever taken more than recommended dose? It was Panadol. In this figure, shows a percentages of respondents reported the sources of information. 84.5% was from family members, with the least was social media, 15.8%. Discussion. The knowledge was significantly different in terms of academic degree and the course of study in the use of OTC. In this study, we found that students in a health science related course is significantly more likely to have better knowledge than those from a non-health science related course. This finding is similar with that of another study in Northwest Ethiopia, where 64.6% of health science students had a good knowledge towards self-medication with OTC. The higher the education level, the more familiar with the clinical practice and medications, which eventually affect the knowledge of OTC use. We found no significant difference between the demographic data characteristic and the attitude. Nevertheless, this study show that very few exposed to unverified sources of health information on social media, which was unexpectedly very low. But the internet was the second majority of the students exposed to the health information as predicted. However, the pharmacist plays a vital role in safeguarding the consumers from potential inappropriate. The risky practice of self-medication with OTC is considered as taking more than recommended dose, failing to check the expiry date, and rarely read the labels. In this study, few of the respondents have taken the OTC medicine Panadol more than recommended dose. The reasons were not effective, the body has adapted to it, unaware of the dosage. In conclusion, only a few respondents have inappropriate practices. Awareness of the proper use of OTC among young adults is necessary to prevent the rise of risky habits in OTC use. These are the references that I used for this presentation. And that is all from me. Thank you. Thank you very much.
All right, thank you, Ishpa. I would like to welcome Prof. Nafiza once again, if you have any questions.
Yeah, hi, Ishpa. Your mic is muted.
Sorry. Oh, hello.
Yeah, very interesting, very practical study, I thought. OTC and the intake of by the public.
Yeah.
Can I ask, what do you plan to do with your findings?
So since this is the first research study on the safe use of OTC, we want to use this as a baseline. The results from this research study will be used as a baseline for the awareness to the public, especially the university students. This is just the first phase. So the next phase will be targeting the older population on the safe use. So we also work with Ministry of Health under the pharmacy department. So we work together to know more about the safe use of the OTC in Brunei Darussalam. Yeah.
Yeah. And there were a couple of times where you mentioned Panadol, because Panadol is the most common OTC drug being purchased by public. Can you tell us a bit why you're worried about overdose of Panadol?
Because there's a study that I found out that if the person take Panadol, like overdose with the Panadol, it will lead to a liver damage to the it will lead to liver damage. As I mentioned about the literature review before, where in Australia they show that there's an increase of liver toxicity due to overdose of Panadol. So which is why I know it's very it is safe to use when used correctly. So we want to know whether whether people in Brunei we want to know if they ever experienced like taking overdose of Panadol. So because they say that most of them say that they overdose with Panadols, which is very worrying because it can lead to in the future leading to liver damage. I mean liver toxicity. Yeah.
Yeah, so that's correct. Overdose of Panadol can lead to liver toxicity or hepatotoxicity. Did it inspire or did it ignite your interest to find out the reasons why they get hepatotoxicity?
What was the question again? Sorry.
Did it inspire you or did it trigger you to find out the basis of hepatotoxicity?
Yes, because from the answer from the students, it triggers me to find out more like what might cause if they take overdose with Panadol. So yeah.
So can you tell us a little bit what you found out?
So far, I didn't research further because I have like limited time to do this research. So I just only found out that that could lead to liver toxicity or I only found out about the liver toxicity only. Unfortunately, I didn't research furthermore in terms of the overdose with Panadol.
Yeah, actually, it's very well established why they get hepatotoxicity when they take overdose, because Panadol is metabolized by a main pathway, and this produces non-toxic metabolites. Okay, so if there's increase in Panadol, they go to a second pathway which produces toxic metabolites, and there's glutathione depletion. I like the study very much. I think it's very practical. That's why I asked you this one, because I think just to be interested in it is not optimal enough. You should be inspired to find out the basis of hepatotoxicity. All right. Thank you.
Okay. Thank you.
Thank you, Prof. Nafisa and the presenter, Ishpa. We'll move on to our fifth presenter by the name of Dr. Kim San Tang from Monash University, Malaysia. Take it over.
Good day, everyone. I'm Tang Kim San from the School of Pharmacy, Monash University, Malaysia. The title of my talk today is The Protective Effects of Zinc Oxide Nanoparticles Against Paraquat-Induced Toxicity. In this study, we are trying to decode the mystery of Parkinson's disease. Parkinson's disease is a progressive neurodegenerative disorder characterized by selective degeneration of dopaminergic neurons in the substantia nigra of the brain, which results in impaired body movement. Parkinson's disease is a multifactorial disease. The main contributor to the development of the disease are oxidative stress and exposure to environmental toxins such as paraquat. Paraquat is a toxic chemical that is widely used as an herbicide, primarily for weed and grass control. Many countries, including Malaysia, have already banned paraquat due to its acute pulmonary and cutaneous toxicity. Despite the ban, some irresponsible farmers still use this illegal poison due to lower costs. Paraquat can induce oxidative stress and has long been associated with Parkinson's disease. Zinc oxide nanoparticles have numerous pharmaceutical and biomedical applications. Zinc oxide nanoparticles exhibit antioxidant properties and have been implicated in the treatment of oxidative stress-related diseases, including cancer and diabetes. Since oxidative stress plays a crucial role in the pathogenesis of Parkinson's disease, we hypothesize that zinc oxide nanoparticles can be used to treat Parkinson's disease. This study aims to examine the protective effect of zinc oxide nanoparticles against paraquat-induced cell death in human neuroblastoma SHSY5Y cells. Here is our study design. The 0-hour time point is when we seed the cells. After cell seeding, we waited for at least 24 hours before we exposed the cells to the following treatment conditions. For the pre-treatment, after 24 hours of post-seeding, the cells were pre-treated with zinc oxide nanoparticles for 24 hours, followed by co-incubation with zinc oxide nanoparticles and paraquat for another 48 hours. For the co-treatment, the cells were treated with zinc oxide nanoparticles and paraquat simultaneously for 48 hours in total. And for the post-treatment, the cells were post-treated with zinc oxide nanoparticles and paraquat for 48 hours after a 2-hour initial exposure to paraquat. All these 3 treatments were terminated at 96-hour time point post-seeding. We then perform an MTT assay to quantify cell viability. These are the results after subjecting the cells to pre-treatment, co-treatment, and post-treatment protocols. First, let us look at the figure on the left, which is the pre-treatment condition. The white bar is the untreated control. The blue bars are the groups treated with zinc oxide nanoparticles. The red bar is the group treated with paraquat only. And the purple bars are the paraquat groups treated with zinc oxide nanoparticles. We found that the exposure to the zinc oxide nanoparticles alone for the concentrations ranging from 0.1 to 0.7 microgram per mil do not affect the cell viability. 300 micromolar of paraquat reduced the cell viability to less than 50%. However, the cell viability was significantly higher by about 10 to 20% for all paraquat groups treated with zinc oxide nanoparticles. Similar trend was also seen in co-treatment and post-treatment conditions. We further examine if zinc oxide nanoparticles exhibit any free radical scavenging activity using DPPH assay. In general, percentage inhibition of DPPH should increase with increasing concentrations of free radical scavengers. In this study, we used ascorbic acid as a positive control. And we found that for zinc oxide nanoparticles, the percentage of DPPH inhibition was close to 0 for all concentrations tested. Just to recap what we found in this study, zinc oxide nanoparticles attenuated paraquat-induced cytotoxicity in SHSY5Y cells. Zinc oxide nanoparticles do not exhibit direct free radical scavenging activity. In conclusion, zinc oxide nanoparticles are neuroprotective as they can attenuate paraquat-induced cell death. They could be a promising agent for the treatment of Parkinson's disease. This study warrants further investigation to determine the neuroprotective mechanisms of zinc oxide nanoparticles. That's all from me today. Thank you and stay safe.
Thank you, Dr. Tang. I'd welcome Dr. Allen if you have any questions.
Okay, so very nice talk, Tang. So just have a couple of questions. So based on your findings, it seems that this zinc oxide nanoparticle has no free radical scavenging activity, right? So
yes.
But yet it managed to protect the SY5Y cells from cell death. So may I know, say, for your model, the paraquat model, how do they induce cell death? I mean, is it induced through apoptosis or due to other mechanism? So that may give you some kind of a clue what would be the protective mechanism mediated by your zinc oxide nanoparticles.
Yes, paraquat is actually can induce oxidative stress, and the cell death mechanisms involved is apoptosis. However, in this study, we did not show any antioxidant properties or anti-free radical scavenging activity of zinc oxide nanoparticles. Perhaps that zinc oxide nanoparticles may induce can neuroprotect the paraquat-induced toxicity via other mechanisms, such as via the zinc ion itself, because the zinc oxide nanoparticles can undergo dissolution and produce zinc ions. And as you know, zinc ions is actually a cofactor for many enzymes in the human body. And one of the enzymes is an antioxidant enzyme known as SOD or superoxide dismutase. And they may trigger that antioxidant enzyme in the cell. Yeah. Right. So our future study is to basically examine the use a cell-based assay to quantify some of the activities of the antioxidant enzymes like superoxide dismutase, catalase, and so on.
Okay, so are you also planning to look at any potential effect by zinc oxide on those, apoptotic pathway enzymes like catastasis and others?
Yes, for sure. Yeah, we are looking into that. Especially we want to quantify whether it's a caspase dependent and or not, because there are various apoptosis process involved.
Okay, that's good. Thank you very much.
Thank you very much.
All right, thank you both. We have now finally arrived at the final presenter for today at this session, NADHIRAH BINTI. Can I have the NADHIRAH BINTI KAMARUDDIN and LEKHA ARUMUGAM? Not sure who will be presenting. I assume it's Nurul. Is it NADHIRAH or NADHIRAH or LEKHA? Anyways, we'll see the presentation now.
A very good day. I would like to greet everyone. Today, me, LEKHA ARUMUGAM, and my friend NADHIRAH KAMARUDDIN will be presenting on in silico analysis of Fortunia caudata active compounds against SARS-CoV-2 main protease, which is known as M-Pro and helicase. Starting off with introduction, COVID-19 is caused by SARS-CoV-2 virus that has been spread rapidly all over the world. There are many vaccines currently available, which includes Pfizer, Sinovac, AstraZeneca, Sinopharm, and Johnson & Johnson. Although there are many vaccines available, this virus has many variants as well as a high mutation rate, which cause it to escape the natural or vaccine-induced immunity. Hence, therapeutics targeting the SARS-CoV-2 RNA synthesis and replication plays an important role in the development of the multi-targeted treatment options for COVID-19. Hence, this study evaluated the binding interactions of Fortunia caudata active compounds and SARS-CoV-2 M-Pro and helicase using the molecular docking approach. Fortunia caudata is a plant that has said having an immunomodulatory and anti-SARS activity against the previous SARS-CoV virus. This plant was used in the study to see whether it exhibits similar effect on SARS-CoV-2. The objective of the study is to study the interaction of Hoitunia chordata active compounds against SARS-CoV-2 main protease and helicase using molecular docking approach based on the lowest free binding energy of the most populated cluster and the binding interactions between the protein and ligand. Moving on to the methods that were used in the study, the first method was ligand and protein preparation. A total of 20 active compounds were selected from Hoitunia chordata and the structures were downloaded from the PubChem. Whereas MPRO with the protein data bank ID of 7BQY and helicase with the protein data bank ID of 6ZSL that were used as targets in the study were downloaded from the protein data bank using the protein data bank ID. The second step was molecular docking. Both protein and ligand were subjected to molecular docking using AutoDock 4.2.6. Lamarckian genetic algorithm was used as the primary conformational search algorithm throughout the docking stimulation. The docking output were presented in terms of free binding energy and numbers in cluster. The interaction between the Hoitunia chordata active compounds and SARS-CoV-2 MPRO and helicase were studied using the protein-ligand interaction profile. Here are the structures of the targets, Hoitunia chordata compounds, and controls that were used in the study. The control that was used for MPRO was N3, and the control that was used for helicase was bismuth potassium citrate.
This table shows the result of the three compounds with lowest binding energy of the most populated cluster for MPRO and helicase. Free binding energy plays an important role to create a higher binding affinity of ligands towards the target. Based on our result, it was shown that the best three compounds interact with MPRO. For protease, these are the compounds shows that to have the lower free binding energy than the control N3, which is caempherol, epsilene, and acetylcysteine. Whereas in helicase, shows slightly higher binding free energy than control itself. The more the binding interaction, the stronger the interactions between the Hoitunia chordata compounds with the target would be. This is the interactions that involve between three best compound of Hoitunia chordata with MPRO. We can see that the interactions involved in the formation of hydrogen bond, hydrophobic interactions, and also few other interactions such as pi stack interactions. Caempherol has a total of 6 hydrogen bond, 5 hydrophobic interactions, and a pi stack interaction, which is the highest in total compared to other Hoitunia chordata compounds such as epsilene and acetylcysteine. This indicates that caempherol has the strongest affinity against the MPRO. This is a diagram that showed the result of molecular docking from molecular analysis. The blue structure represents MPRO and the golden orange structure represents caempherol. It shows all the interactions that form between MPRO and caempherol. Based on this result table, the control for helicase, which is bismuth potassium citrate, shows to have the strongest binding affinity and the highest interactions involved with helicase compared to other Hoitunia chordata compounds such as saprophyrin B and positrene. Thus, it shows that bismuth potassium citrate has the strongest binding affinity against helicase. These are diagrams from molecular analysis that shows the difference in interactions involved between bismuth potassium citrate and saprophyrin B with helicase. There are more interactions forces involved in helicase control structure compared to saprophyrin, which makes it to have the strongest binding affinity towards the target. Based from the result we obtained, caempherol, epsilene, and acetylcysteine are the most potential compounds for MPRO. To support this result, previous studies had shown that caempherol, acetylcysteine antiviral effects towards SARS coronavirus and also cytomegalovirus by suppressing the viral genes, whereas some alkaloids have antiviral effect against herpes simplex virus type 1 by interfering the viral replicative cycle. Quercetin, which is a flavonoid, it inhibits influenza virus by reducing mRNA transcription in dose-dependent manner, and it inhibits rhinovirus by blocking the endocytosis via P1-3 kinase inhibition. All these interactions involved in protein and Harishini compounds are necessary for the protein folding and stability, and also to remove all the water molecules involved. Hence, these interactions plays an important role on the free binding energy and also for the binding affinity. Based on our research, we conclude that flavonoids and alkaloids might bind against SARS-CoV-2 and pro-enhance inhibition. Research, we and also for the binding affinity. Based on our research, we conclude that flavonoids and alkaloids might bind against SARS-CoV-2 and pro-helicase. However, further research studies might be needed to support our findings. These are some of the references involved in this study research. Thank you.
All right, thank you. That's interesting. We have two presenters, Nur Nadhirah and LEKHA ARUMUGAM. Both are here with us. Prof. Nafiza, any questions?
Yeah, no wonder there were two voices. So I was wondering what was happening. Anyway, a lot of work done, very interesting work. Either one of you can answer. In a nutshell, or if somebody were to ask you, what is the headlines of your study? What would you say? One headline.
Hi, Doctor. It's a nice question. I would like to sum up by saying this could be, we could name it as SARS-CoV-2 potential compounds.
Yeah, but if I always believe that researchers, before they embark into the study, they would do a literature review, right? And they would read around the study, okay? And one thing that is going about on social media and in articles now, and you hear it every time on the news, is ivermectin. Can you tell me about how ivermectin, how it acts? How it is supposed to act, because some people say it is it's effective against coronavirus.
Yes, doctor. This one, when we were doing our research, we were asked to find for our control. Control means so that we can compare it with another with our other potential 20 compounds that we used here. So while we were searching for the compounds, we found out that ivermectin could inhibit the SARS-CoV-2 helicase. So during that time, they said that, but no further studies has proven. It's just what was in the research mentioned that it could be one of the potentials.
Okay, what do you think is to that? I know there's not much studies. Maybe you couldn't find a lot of studies, but if you Google on YouTube about the mechanism of action of ivermectin, I think there's one very short video, but very concise and very clear, and you would then know how ivermectin acts. But whether it is, of course, we know the basic science is that, but of course we would need clinical trials, right? Okay, what would you say is the strength of your study?
Our study, we could say it has a higher strength because it was also proven that one of our compound, chimferol, was actually used for our previous SARS-CoV that came before this. So we actually have a doubt, high curiosity level, whether it could show the same effect to this virus, since they are from the same family, but just they are SARS-CoV-1 and 2. So because of our curiosity, so we want to see whether the same compounds could have the same effect on this virus, because they are the same family. So maybe some genetics as the similar thing could potentially block their actions. Yeah. So that was our curiosity. So yeah. So our studies, we feel like it has a good strength.
Do you think there are any limitations to the study?
Yes, there's limitation because this is a computational study actually. We have not performed yet an experimental study. And these results were actually chosen from the aspect of molecular docking, where they said there are scoring functions that will help us to find out the binding affinity between a ligand and compound. So from that, we find out that in terms of AMPRO, we find out Chimeferol have the strongest binding affinity because it has the lowest binding energy from the scoring function. So in terms of helicase, we find out that Sapphirodione B has the strongest binding affinity from the free binding energy that it has, which is lesser.
All right, thank you. No more questions.
Thank you, Doctor.
Right, thank you very much, both of you. With that, we've come to the end of our oral presentation session. I would appreciate if all the presenters today would turn on your camera so that we could take a group photo along with our two distinguished judges and committee members as well in the panel room. Vanessa, can you count?
Yes, okay. So everyone smile. 1, 2, 3. Wait. Okay, again. 1, 2, 3. Okay, thank you, everyone.
Thank you, everyone. Don't forget to join in our poster presentation session at, I believe at 4:15 p.m. There are two rooms, rooms 1 and rooms 2. Poster presenters can already join in and raise your hand so that we can promote you to the panel room. All right, have a good tea break and we'll see you guys soon. Well done, everyone. Thank you, Prof.
Thank you.
And Dr. Ellen.
Thank you.
Thank you.
Thank you, everyone.